Okay, almost done. We are on Unit 6, page 17. And we are going to be doing transcription, RNA processing, and translation. So there's a lot going on at the same time. I'm going to try to break this down into baby steps. I'm going to be using Legos. It's going to be okay. Y'all are going to be able to figure it out. I completely believe in every single one of you. First, we have DNA. And that DNA is going to undergo transcription in order to make messenger RNA. So the process is called transcription. We're making messenger RNA. There's 3 different types of RNA. And that messenger RNA will undergo translation to make protein. All right, so the DNA making a copy of mRNA, this is transcription. This happens in the nucleus. Because the DNA does not leave the nucleus. DNA is on house arrest forever. It will never leave the nucleus. The messenger RNA is able to leave the nucleus, so it will take the message of the DNA out of the nucleus into the cytoplasm where translation can occur. So the mRNA will leave the nucleus and will go to the cytoplasm where it will undergo translation to make protein. Alrighty, so let's say that we have our, let's see, I'm going to bring some pieces over here. Okay. And by the way, this is very different from DNA replication. We're not making another copy of DNA. The purpose of this process is to make protein which will perform some sort of action in your body. Alrighty, I have all my pieces. There we go. So, let's say we're in the nucleus. We got nucleus right here. Let's say that the nucleus is in a cell. Cell, boom, there we go, we have a cell. Nucleus. Here we have a cytoplasm. So, we have the DNA. Let's say that this right here is the DNA, just chilling in there, right? And the DNA is going to open up to make some mRNA. The mRNA is going to leave, and then it's going to disassociate from the DNA. It's going to leave, and it's going to be floating around with the intention to leave the nucleus. Before it leaves the nucleus, it undergoes some processing. So this mRNA is going to be processed. It would have been easier if I used this. But it's pink, so it's fine. So the mRNA, once it's made, it's going to go over here, it's going to get processed. Parts of the mRNA that will not be used will stay in the nucleus. The parts that will be used to make the protein will exit the nucleus. So these are the exons, this is the intron. I will re-explain this as we go along. So don't worry, I will re-explain with labels. And then what we're going to do is we're going to add on a 5' cap and a poly A tail. Let's see. There we go. 5' cap and a poly A tail. And then boom, there we go. That is going to be the mature mRNA. The mature mRNA goes out of the nucleus through a nuclear pore to the cytoplasm. At the cytoplasm, it will go to a ribosome. That's the ribosome. At the ribosome, there are 3 parts of the ribosome. There is a part where the you have entry of the amino acid. You have the middle where the protein is made. And then you have the last part, which is the exit. So amino acid, protein, exit. APE. So this right here, this goes in here and is read through like a zipper. And as it is read through, it's read through head first, and tRNAs, transfer RNAs, will come down. So let's say that these are the transfer RNAs. There we go. And this one too, why not? They're going to come down and they're going to have little amino acids attached to them. There we go. Boom. There we go. There we go. There we go. So these are the tRNAs. They're going to come down. And imagine there are 3 slots here. So in order to make that easier to see, I'm going to see if I can get a Lego with 3 little pieces on it. Okay, boom. Amazing. There we go. 3 little pieces. There's 3 slots. So what happens is that the mRNA goes into the ribosome, and at the beginning, that's where the amino acid comes in. So here we have our tRNA dropping off the first amino acid. Boom, there we go. That's where the amino acid goes in the first slot. And then what happens is everything moves down, swoosh. Everything moves down, swoosh, like that. Moves down a little bit. And then the next mRNA codon is going to be read. The ribosome is basically reading this like an instruction manual, like a sequence. And then the next mRNA codon will be read. And then the tRNA will come down with the second amino acid, swoosh, just like that, in the second slot. Or the second one coming down. And then the middle one will be where the protein is made and elongated. And then this blue one now will exit. Bye-bye. It's going to be empty. There we go. And then the next amino acid on a tRNA comes down. It's going to go here. Everything's going to be shifted down. And then this amino acid is going to attach on to the existing amino acid chain. There you go. So these tRNAs that are floating around, they are going to be able to go and grab another amino acid. Just like that. And then once the end of the mRNA is reached, the amino acids dissociate. They are in a chain, so that the chain will clump up and then they will form the protein. So that was kind of like a high-level overview of what's going on. Now I'm going to go into it in more detail with labels. All right, so here we have our DNA. Our DNA. DNA. The DNA, let's see if I can use a better marker for that. Let's use this one. DNA. The DNA opens up. And DNA has codons. So, for example, let's say that this is a sequence, and each of these buttons is a codon. Let's say T, A, C, A, G, T, C, C, C. Let's say that that's what's going on up here. This will be one codon, so that would be like one button. This would be another one. That would be another codon. And then let's say AAA is at the very end. AAA, that would be the next one. These codons are going to represent an amino acid. They code for an amino acid. So when the mRNA comes down and gets coded, it will be coded on one side of the DNA, not the other side. One side will be the coding side, the other side will be the non-coding side. Or this side will be the complementary side, this side will be the coding side. Again, this side is the complementary template DNA that makes the mRNA. This side will be the coding strand. Because this side is complementary to this, and this is also complementary to this, this and this will be basically the same thing, except this will have T's and this will have U's. You can go to page 18, and that's where it talks about the template strand and the coding strand. Again, this is the template strand. This is the mRNA. This is the coding strand. So 1, 2, 3, 4, because we have 1, 2, 3, 4 codons. So this would be like the mRNA codons. The mRNA is made. The DNA closes up again. So now this is the mRNA. This is the pre-mRNA. It needs to undergo processing. So when it undergoes processing, it then will become mature mRNA that's ready to leave the nucleus. When it undergoes processing, what happens is that introns stay in the nucleus. So introns, like this one, will stay in the nucleus. We have 1, 2, 3, 4, but we only need 1, 2, 3 codons. Introns stay in the nucleus. And exons, which are these, 1, 2, 3, will exit the nucleus. So exons are able to exit the nucleus. And then we're also putting on a 5' cap and a poly A tail. So this will be the poly A tail, and this will be the 5' cap. So what makes an intron and an exon an exon? Well, I mean, theoretically speaking, any of them could be introns, any of them could be exons. It depends on what type of protein you're making. This one mRNA strand could be responsible for making like 4 different types of proteins. So it really depends on how you want to splice things up. It really depends on like which of these you want to keep. Let's say you only wanted to keep 2 of these. Boom, there you go. Then those are the exons, then those are the introns. Let's say you wanted to keep the second one, but not the first one, stuff like that. So let's say you wanted to keep everything but the last one. You know, you can have different combinations of exons, and with the different combinations of exons, that can make different types of proteins. And this right here, the exons that, you know, you can have in different arrangements, you know, of which ones you're keeping, that is called alternative splicing. So alternative splicing. Again, alternative splicing is how one mRNA can make different proteins. The mRNA will leave the nucleus and go to a ribosome. So let's say that we have a ribosome which has 3 parts. The A part, the P part, and the E part. The A part is where the amino acid first enters. So here we have our tRNA. And the tRNA is transfer RNA. It's going to carry the amino acids. These right here, these are the amino acids. They're going to carry it to the A part. That's where the amino acid enters. As the codons on the mRNA are being read. So let's say that we have TAC, AGT, CCC, AAA. The code that would be complementary to that would end up being it would be U. There's a U now. AUG U C A G G G U U U. Because when we're going from DNA to RNA, the T's are now U's. So each one of these is an mRNA codon. Each one of these is an mRNA codon, which is made of 3 letters. Each of these is made of 3 letters. And on the tRNA, each tRNA has an anticodon. So at the bottom of these is an anticodon, which is complementary to an mRNA codon. So let's say that the first one is going to be AUG. So this one right here is going to be AUG. That's how we got it, AUG. Then AUG is going to be complementary to the anticodon UAC. So that's how you know exactly which tRNA is going to go down. It's going to be the tRNA with the anticodon UAC that's going to go down. And the tRNA with the anticodon UAC will only carry the amino acid called methionine. And methionine will always be the start codon, or like the start. So AUG will always start an mRNA, and AUG will always be coding for methionine, and methionine will always be the first amino acid in the chain. So this one would be methionine. And then it goes to the P side in the middle, and the P is for protein. That's where the protein elongates. Another tRNA will come down with amino acid. And then we have in the elongation of the protein in the middle. There we go. And then the empty tRNA will just leave, go bye-bye, probably go get another amino acid to come back. And then let's say that another amino acid comes down, there we go, and brings down another amino acid. There we go. Then this moves down, this red one goes away, and then the code, you know, this will keep moving down. And then boom, the strand just keeps getting longer and longer. Let's say another one moves down. Let's say that it is this green one. Let's say that this is another tRNA. tRNA goes down here. Everything moves down a little bit. The tRNA is carrying this amino acid. This amino acid is going to be added to the chain right there. This orange one will just leave and exit off the E side. Let's say another one comes down with another amino acid. So this is another tRNA with another amino acid. Let's say it comes down to the A side again. There we go. This green one will exit on the E side. And then this will slide down, and then the chain again elongates. Okay, so let's say that we reach the end of this. Well, everything disassociates. This becomes our amino acid chain. It balls up, and then boom, you have your protein. And it may be functional now, or you may need to do some post-translational modification. And if you do the post-translational modification, that means you're probably adding some things to it. For example, a methyl group or a hydroxyl group, and the extra additions will help it become more finalized so that it can go on and do its job